What drew me to this project of studying endocannabinoid signaling is mainly due to the increase in legalization, recreational use, and medical use of cannabis, and understanding the physiological effects that cannabis use has on the body and how the endocannabinoid system normally works is becoming much more important. And we really don't know everything about endocannabinoids. They aren't fully understood yet, such as how does cannabis use impact normal physiological functioning, and is it really harmless, or do we just not know enough about the system and the long-term effects of widespread cannabis use? So understanding the natural signaling of the endocannabinoid system will help us to more fully realize the physiological implications of its use and to help develop drugs that could be used to treat some of the medical issues that cannabis is currently being used for. So, how does cannabinoid signaling work? Well, it modulates neurotransmitter release in a retrograde fashion. So, to understand how it works, we first need to look at regular neuronal signaling. In the classic model of neurotransmitter release, the presynaptic neuron will receive a depolarization signal, which will cause the release of neurotransmitters from the presynaptic neuron. These neurotransmitters will travel across the synaptic cleft where they will bind to receptors on the postsynaptic neuron. This causes an influx of calcium, which causes depolarization. With retrograde signaling in the endocannabinoid system, neurotransmitter binding to the receptor causes an influx of calcium. This activates phospholipase C, which then will create 2-AG, or through phospholipase D, it will create anandamide. Once the anandamide in 2-AG has been released, it will travel across the synaptic cleft where it will bind to CB1 receptors on the presynaptic neuron. CB1 receptor activation causes an inhibition of adenylyl cyclase and calcium channels and activates the MAPK pathway. The result of this signaling is hyperpolarization of the presynaptic neuron, which inhibits neurotransmitter release. This whole process is known as retrograde inhibition and is a hallmark of endocannabinoid signaling. So with the complexity of endocannabinoid signaling, what sorts of experiments can we do? Well, one area of interest is in the endocannabinoid system's ability to modulate hunger, and so we can do a feeding study to look and see how cannabis use affects body weight. So cannabis use is known to cause the munchies, which is an increased appetite for highly palatable foods. So we can see how does this affect overall body weight and how does it affect the presence of various hormones that cause this increase in feeding behavior. So for our methods, we will repeatedly treat mice with cannabis and give them access to food ad libitum, so they're able to eat as much as they want. During this, we will measure their body weight and their food intake over time to see if it changes. We will also measure the hormones that control food intake, such as leptin and ghrelin, to see if this changes over time. This will tell us if the hormone concentrations and food intake are different between the control mice and the mice treated repeatedly with cannabis. Now to look at signaling, we can look at the relative concentrations of the endocannabinoids in the brain to see if that changes, and we can look at the amount of receptor expression using a Western blot to see if that changes over time too. We can visualize this using immunolabeling and confocal microscopy to see if the distribution of the cannabinoid receptors changes. So what will this tell us? Well, it will tell us if cannabis use changes normal endocannabinoid functions. So does it change other endocannabinoid-related pathways? Does cannabis use cause changes in body weight with normal feeding, and does this have implications for weight loss? And does cannabis use change receptor expression localization, and could this have implications for dependency?